So we're on the theme of processing. We're moving on to segmentation and subtomogram averaging, led by Jake Johnston. He's actually a grad student here, and he's one of the stalwarts at NYSPC, really trying to dive into that spaghetti of processing. Jake, what's your favorite part of tomography work? Or instrumentation?
Probably just processing.
Processing in general. Okay.
It's on a computer chilling.
Okay. Take it away, Jake.
Okay, yeah. So the title of this talk is segmentation and subtogram averaging pipelines. So I'm just going to try to provide a brief overview of the different pipelines, things like that, because you guys have next pipe that you just had, and then Alistair's going to talk about 4 clinics. So I'll just try to give a good overview here as much as I can. Yeah. So one thing I'm going to talk about at the end is approaching different biological questions, or at least one of them, using either subtogram averaging or segmentation workflows. And then I'll also discuss, just kind of an overview of several software pipelines. So this is something that's always constantly changing in CryoET. So and it's kind of difficult to catch up with sometimes. So just to kind of provide a brief recap of CryoET workflow summary. So generally, you kind of have your EM grid, right? You guys have probably seen this from different sample preps. You put a extracellular matrix on there, for example, if you're punch freezing. You put your model cell system on there. Ideally, the model cell system attaches. You have a fluorescent tag for the region of interest that you want to target. And this is kind of what gets into cryoplan. Ideally, if your sample's 350 nanometers or less, 500 is a little too thick. You put on Rios, collect the tomography data set, basically a tilt series. You can actually back project that into a 3D volume or a tomogram. And then from there, you can actually take the tomogram and then do a downstream analysis. And that's usually involving a subtogram averaging and segmentation pipelines. So once you get a tomogram, that's kind of where a lot of the work comes in, a lot of time comes in. Is figuring out how to analyze the data and what kind of workflow you want to use for your biological question. So this is kind of a workflow for it. So you collect your data set, and then you basically have a set of frames for each tilt. You do motion correction on those, and then you get a per image. You get basically a micrograph per tilt, per image CTF estimation, tilt series alignment, program reconstruction, particle picking segmentation, subtogram averaging, alignment, classification, per particle tilt image refinement. Then you can actually take your maps and then put them back into the tomograms for cellular context. And then you can call that by a statistical analysis if you need that for biology. And then hopefully you publish at some point. And so this is just kind of a little slide here showing the different steps, and then just the different software that's available. So Misha talked about Cilium Tomo 5. Generally for motion correction, CTF estimation, you can use Motion Core, or NextPipe has this all implemented as well. Tilt series alignment. So this is kind of showing really just how many different pipelines there are in tomography workflow. There's a little link here. Actually, I found on GitHub that has like an isolated workflows for each step there. So that was kind of cool. And one of the pipelines you do after motion correction or denoising is segmentation, followed by from averaging. So these are like the full pipeline here for subtilt refinement. So you can kind of take this, do segmentation, and also do averaging. So motion correction. So your frames, you want to look at what kind of motion correction you want to do. Generally, it's usually Motion Core 2 or 3. That's pretty robust. Or warp. And so all these will do global or local alignments. They're pretty fast, so it's pretty quick off the scope if you're doing it on the fly. And they implement dose weighting, so you're taking into account the different doses on each frame. Motion Core and warp, they're all GPU accelerated. And then Motion Core 3, I believe, has CTF estimation as well. that's useful, but Warp is generally pretty good for on the fly, at least the Windows version. You can figure out a copy from the scope. Yep, and then Warp actually implements an additional step, 3D modeling, motion projection. But yeah, Alistair probably covered this pretty detailed later. So you get your frames, you align your frames into micrographs. Ideally, the data is good, and you get CTF estimations that are about 3 to 4 angstroms. And so Warp will actually implement local CTF estimation, tilt axis, and it'll actually give you a correlation value for the defocus handedness of the tilts and local CTF estimation. You can also use CTF fine, but Warp's actually kind of nice because it does give you whether you have the correct defocus handedness of the tilt series. Okay, so you get your tilt stack, and then you have tilt series alignments. So a lot of times you'll see fiducials in there. That's for tracking the tilt series to align it. And so kind of the problems with is getting sample preparation, getting well distributed feeds. Some workflows are kind of semi-automated. A lot of them are automated now. One of the things that came out that was really useful was RTOMO. So it uses a similar algorithm to ProTOMO, which is kind of a projection method to align it. And it's fast, so it takes like a minute or two for tilt series. And so probably one of the critical parameters for actually aligning tilt series with RTOMO is adjusting the line Z value. So I left that on there for sharing. And that depends on basically the sample thickness for the matching. And some other softwares like iMod things will use path tracking. So probably one of the kind of annoying parts of tomography processing is actually generally annotating your data. This can take a lot of time depending on what your data is. So like particle picking, segmentation. So for particle picking, you can do geometry-based approaches. So if you know the geometry of your sample, you can use that to your advantage. Template matching is something that's been playing around a lot. Things like Python, stopgap. A lot of deep neural networks for particle picking. And then kind of iterative approaches. So you get a set of particles, train, do downstream processing, and then reuse that for training. So this kind of goes back to line with machine learning and potentially template matching if you get a map that you're interested in using. And there's always manual if you want to do that. So, and one of the things I like to do for everything for picking is actually denoise the tomograms. And the reason for that is because a lot of time you have things in there you can't really actually see without denoising. So one of the things I did here a couple of years ago was actually look at data set on just purified hemoglobin. So if you look at the tilt series, you literally can't see anything. And then just a weighted back projection on the tomogram and deconvolution. You really can't see anything, but if you denoise it, you can actually see all the hemoglobin particles on there. So it's pretty drastic change. So I'm trying to pick something up that, right? So, and then the other thing that came out about 2022 was IceNet. So this actually does a missing wedge correction. And so that's what's showing here is actually a VLP. So if you look at this plane here, you can see that you have the missing wedge streaking. And then actually we'll come in and fill in the missing wedge, and then you get clear particles for picking. And this is purely just for picking purposes. Another example of this is just on a Empire data set, 10499. So labs have shown just a deconvolution and then writes the actual missing wedge correction. So you can really see like the ribosome particles and things like that. And I put the GitHub on here too for these. So it's definitely worth using. Takes a few days to train, but it's definitely worth it. And then, yep, so there's some different tomography particle picking, segmentation pipelines. One of the kind of older ones I think has been EMEN2, where you kind of select the positive and negative examples for what you want to segment. So there are softwares that came out. SMLC here just put out TARDIS, which is a kind of automatic segmentation for filaments, membranes. Membrane seg is also a good one. That one gives you initial segmentation. And yeah, I put the GitHubs there too. So I'm big on the neural network stuff here. And then of course there's always a dynamo you can go back to. So dynamo is really good for using geometry of the sample. So things like filaments, you can actually use it to pick, trace, it'll use geometry for it, and then that will help you with downstream processing for averaging. You can use it for also membranes, so you can actually create meshes around your membrane if you want to, and you can actually oh, it's like frozen. Okay, sorry about that. Yeah, so the internet. Yeah, so you can actually use enamel to create meshes around the membrane, and then use the meshes to actually pick particles on the membrane. So some of the things that are kind of useful, like membrane seg. For example, you can actually use that segmentation to search for particles on the membrane. And I think that's kind of a really useful case of mixing deep learning with geometry. Another case, picking vesicles. So if you guys ever did the old Team Tomo tutorial, I think it's still there. You kind of go through, you can actually pick spherical particles there. Use the lasses to actually average. So yeah, I mean, Dynamo is full of a bunch of geometry based processing. Yeah, and of course there's a template matching. So you have your tomogram, you get your map, and you essentially just use a bunch of cross correlations, Angular searches, try to match your template to particles in the tomogram. So there's been a lot of software packages that come out for this. SoftGap, Python, all became GPU accelerated, so they have a faster way of searching for angles. And yeah, it's just an example of a Python particle picking. So it's worth trying if you can optimize it. Yeah, and it's all GPU accelerated now. Yep. And then the thing I used to use a lot for particle picking tomography was Cryo. So one of the things I've had with Cryo, so actually you can pick. See here. Doesn't look good. I think internet stuff. Okay. Yeah, so I've actually used Cryo a lot. So with this software, it actually trains on 2D slices of the tomogram. So when you actually train it, you're supposed to pick every single slice of your particle and then use that for training. If you get creative enough, you can actually write a script to just generate those slices for you. So you just pick once. One of the things I've had with it is that it works okay, but it tends to underpick a lot of things, but the picks are generally clean enough. Yeah, so I think there's even more. There's a bunch more other pipelines too, like DeepFinder, things like that. Like it's almost impossible to cover all of it. And then you get into subtomogram averaging, right? So your particles, you extract subtomos, and then you want to look at averaging, right? So you're going to take these subtomos, average. And these are some examples of pipelines you can use for it. Things like eman2, rely on, has been doing this for a while. EM Clarity, it's kind of been out for a long time, but not super stable. Dynamo, you can go through to Warp, which technically is actually through Relion, so but that pipeline, and then of course NextPipe, which you guys have seen through the tutorials here. So one thing's about subtomogram averaging, if you just do 3D alignment, you can't really refine the image angles, but if you go back to 2D alignments, you can actually refine those image angles. You get higher resolution. And a kind of a common workflow for a lot of these subtomogram averaging pipelines is to heavily bend your tomogram, so something like 10 angstrom per pixel to 16 angstrom per pixel. Pick those coordinates and then bend, do the alignment so it's faster, because a lot of these alignments are actually done with 3 subtomos, right? And that tends to be computationally heavy. I think things like the line 5 tomo uses what's called like pseudo subtomos, and that goes back to the 2D 2D alignment. So that can be kind of useful for like limiting, lowering the amount of resources you need for alignments. But yeah, the general strategy is usually just to take bend, align, unbend more, align, and just kind of start optimizing your poses. I didn't get into too much detail about classification, but you can align and then just classify kind of similarly to what you would do in single particle. And yeah, this actually can be pretty slow if you have a lot of subtomos. So I had a data set that was 110,000 subtomos, and that took probably 4 or 5 days just to classify. So that can be kind of a slow pipeline, actually, and that seems to be where I run the ball next these days. So what's really useful is subtotal refinement. So these are all software pipelines that actually implement this. So you actually go back to the TIL series and then redo the alignment so they're better. And I have an example of this I just did about a week or two ago using Warp Linux. Just to show you like the difference in resolution. So yeah. So it's actually using the Empire 10499 data set from the original Warp N paper. So just with general subtotal averaging, you get something like a 10 angstrom map. You can do a M refinement. And yeah, Alistair will tell you about this, like doing stepwise and things like that for optimizing parameters. But this one round of M, you're gonna go from 10 angstroms to about a 5 angstrom map. Second round, 4. Third, about a 3.8. And then a couple more rounds, you get us to about 3.69 pretty easily. More classification to get rid of some subpar particles. You probably could push this to what paper resolution is, which is like about 3.5. And yeah, you guys will probably spend all day doing this stuff. So, okay, so you get your high resolution map, and you're interested in the biology, right? So you want to see where these particles are aligned in the tomogram, how they are related to other parts, like where you segmented. So there's actually software you can use, which is R2X. Interesting. Yep. Yeah, it's actually a little VR implementation too, which is pretty fun. Yeah, so it's actually viewable in Chimera. So you can actually take your SAR files, your poses, place them back into the tomogram. Also takes segmentation files, and you can look at it and go, okay, this is how my particles are arranged. Are they localized in certain areas? Yeah, and like I said, it has this kind of like a little VR, so you can actually put these in VR and manipulate them. So yeah, that's definitely worth doing if you, I guess, maps and want to place them back. Okay, so yeah, one of the things is there's a lot of software in tomography, and so like how do you place different software to the data sets? So, and it really depends on the biological question you want to answer. Do you need averaging? Do you need segmentation? You need both. That really affects like how you collect data, pixel size, mag, things like that. So I'll just kind of have some slides on something I did recently, which is looking at migrating cells, mostly a segmentation based project. But I'm not going to go through the biology too heavily because it's already 20 minutes in. But yeah, so basically migrating cells, they have a bunch of different functions. So they're involved in embryonic development, wound healing, the immune system, where they actually migrate. And like embryonic development, it's actually important for these to actually form different organs, things like that. And so one of the actual characteristics of migrating cells is that they actually have a polarity, meaning that the front and the back structure of these cells are different. They have a gradient. So essentially, the front will have these branch actin filaments, the back will have these long range non-branching filaments, different signaling pathways, things like that. So it's actually a pretty cool aspect of this. And so for this project, we were kind of looking at how these membrane contact sites actually differ between the front and the back of the cell. And so these membrane contact sites for biology purposes, like so like things like the ER will actually form contact sites of different organelles. This is important for like a bunch of processes, but so you have two different types of ER. You have like a rough ER or sheet-like ER, and then a cortical ER, tube-like. And it's considered a contact site when they're within 0.5 nanometers or so. And so we were kind of interested in how these are organized. And there's functional diversity in these. So there's different proteins that transport things like calcium across for signaling pathways. There's actually other proteins that mediate this. So biologically, it's pretty important. So I have this collaborator. Basically, has a construct that resides in the ER. And then when it's within a contact site, it'll actually fluoresce and form what's called like a puncta. And so they actually took these, put some cells in some slides, looked at the migration, And so where this fluoresces is where most of the contact sites are. So this is actually the back of the cell. So they're seeing more of these punctas at the back relative to the front. And then they're kind of like, okay, why are we seeing more contact sites in the back relative to the front? So they kind of assumed it was related to ER curvature gradient. So how ER is actually organized. So they had a couple markers here, clip 63 for ER sheets, and then ER tubules for RTN4. So what they actually saw here is this is cell migrating, that there's actually more clip 63 or sheet-like ER in the back relative to the front. And then if you look at this here, they actually saw the opposite. So they're seeing a lot more sheet-like ER back relative to the front. And so kind of getting into what you could do this with gravity was this is cell biology, so they have no idea how this actually looks structurally, right? So basically, how would you compare this with front and the back using cryo-electron tomography? And then obviously with this method, we can actually see high resolution. And then I'm sure, I think one of the initial talks talked about micropatterning, but you can basically force these cells to migrate by putting linear stripes. And this actually, the geometry of this actually forces them to migrate. This is just an image of them on Zeiss. These are cells that were micropatterned, stripes, and then this is actually nice E-stack here of those cells. And so this is kind of where on the data collection aspect this comes in. So we were only interested in mostly looking at the organization of the ER. So we didn't really focus on symptomatic averaging so much. And the other thing was the morphology of cells were so different between that front portion and the back portion that we can actually just target these without actually using clamp. So that prompted kind of a lower mag data collection. So like something like 8 micron to focus, and then something lower pixel size. So it's like 4 inches per pixel. So not really focused on symptomatic averaging this guy. And so that's actually the front of the cell. And this is actually an isonet corrected tomogram in the front. And so you can see why I like doing this method, because you can really see all the filaments, everything's very, very clear. And so that really helps out with a lot of the segmentation pipelines. It's kind of cool. So this is kind of one way of segmenting it. You can actually take initial segmentation from something like TARDIS, filament seg PV, maybe membrane seg. You can actually import that into Dragonfly if you want. Train them 5 frames, just clean it. This is kind of uses a unit layer architecture. So you can actually take these frames, train it, and it'll actually predict the segmentations of the whole tomogram. You can iterate this if you want, and then you can finally clean the final segmentation. This actually came from a little BR paper we did here. Kind of cool. Yep. So you get something like this. Oh, that's not actually. So in red is actually the plasma membrane using a pipeline like this, and then in green is actually a ER at the front of the cell. And you can generate movies like this in Dragonfly if you want. So one of the cool things you can do with tomography. And then, yeah, this is the back of the cell, so you can actually get and this is actually kind of a thicker tomogram, so it's like 250 nanometers or 300 over carbon. And isonet corrected. You can really see all the ribosomes. You can see all the ER. Granted, it's like 8 micron to focus, so if you try to averaging those, that's not going to work out too well, but they can generate a whole sheet like ER in the back. Again, green is ER, and then red is actually the plasma membrane there. So one of the things you want to do this was actually characterize this, right? So you get your so your segmentation, right? You're not really done there yet, because you want to quantify things from the segmentation. You want to look at something like the distances between membranes or the curvature of the membranes. One of the pipelines you can actually use is a circuit form metrics. So this is written by Ben Broad. It's actually very useful. So one of the caveats is that once you get the segmentation, you have to make sure that what you want to look at is actually separated or labeled differently. Let's say like inner, outer membranes of mitochondria. But this is actually a pretty useful pipeline. It's like 4 scripts. There's a little bit of scripting for the back end. There's the statistics. And it basically converts voxel-based segmentations into a surface mesh. That's what's shown there. So using this pipeline, one of the things about the ER is that the front should have been more curved than the back because tubular ER tends to be more curved. And so one of the pipelines used was actually, it imports a library, PyCurve. Uses a mesh triangular, uses a triangular geometry there. And you can actually use this to calculate different curvature values, distances. So I actually use this to quantify basically the front ER from the back ER in a way that was just measuring the curvature of it. And of course, the front has more curved ER. Another thing you can do with this, for example, is this is an example of calculating distances between ER luminal membranes. So to do this, basically, you would take your full segmentation, separate. This is a separate membrane, calculate the distances between those, and then do some downstream statistics. So it's actually a Matt Whitney test here. And they can actually, so in this case, we saw, the front ER was had a short ER luminal distance, which we expected. But this is just an example of how you can actually use CAR-IT to look at the different morphologies there. That's probably one of the cooler results. So one of the things we saw was actually the organization of the contact sites in front and back here. So this is actually the front contact site, just forms a small area, and then this is the back, which is the sheet-like. So using this kit, you can actually calculate the total area within a certain distance. In this case, 40 was the cutoff, and calculate the total surface area that was there. So that's kind of really the power you can do with just segmentation using these pipelines at the back for quantifying these different structures. And then, yeah, it's just an example here. So we measure the distance between the contact sites. The front was further plasma membrane. This is tighter, and that's 12-fold more area. More biology stuff there, but yeah, this is more of an example of just trying to show what you can do with it. Okay, let's do that. Not sure if there's much questions about any of this, but there is. Yeah, thanks.
We have
Yeah.
they have the goal set on these leads.
Grow them? No, actually just put them on there. Like yeah, they weren't growing on there too long. It was like a hour, 4 hours or so.
Oh, okay.
Yeah.
Something.
Yeah.
Yep.
Yep, on the microbiome grid. Yeah.
I was curious, you mentioned that we're trying to classify 100,000 modules.
Mhm.
And you mentioned that it took 4 to 5 days. Can
Yeah.
you say something about what kind of resources we're using for that?
This is on 4 A100s.
4100.
Yeah, with Reliant 4. So
What
yeah,
was the box size?
the box size wasn't too large. I think this one was probably actually 64 to 128, if I remember correctly. Box size.
How many classes?
This was quite a bit. So we're really trying to filter out junk in this for a different project, but it was 50 to 100 classes, up to 500 even. So a lot of classes. Yeah.